Hello students, welcome to 7th lecture of surface chemistry. In previous lecture our discussion was about catalyst, and in this lecture we will know about catalysts more. Today's first point is shape selective catalyst. That means these type of catalysts do not depends on the chemical reactivity, but rather it depends on shape of molecules. The catalytical reaction that depends upon the pore structure of the catalyst and the size of reactant and product molecules is called shape selective catalysis. Zeolite is a good shape selective catalyst because of their honeycomb like structure. So zeolites are usually stones made of aluminosilicate compound. That means it contains aluminum, silicon and oxygen. So as mentioned here, zeolites are good shape selective catalysts because of their honeycomb like structure. In these few examples we cannot see these honeycomb like structures because their structures are microscopic. That means we need to zoom in the images, otherwise it will look like normal stones. It has many different type of shapes and many different type of composition. So sometimes you may see these type of stone like zeolites, or sometimes these type of crystalline zeolites. All of them are aluminosilicate compounds with different type of arrangements. So as mentioned, they are microporous aluminosilicate with the three-dimensional network of silicates in which some silicon atoms are replaced by aluminum atoms, giving ALOSI framework. That means, as we know, silicon belongs to carbon family, so usually it forms 4 different bonds, while coordination number of aluminum is 3, so that's why it will form 3 bonds. So as the amount of aluminum varies, you will get different type of structures from the zeolite. So here are few microscopic representation of zeolites. As we can see in this image, you will see different type of pore size or holes present in single layer of zeolite. This is for example, this is single layer of zeolite, and actually it will be something like this. That means multiple different layers of these type of plates will be placed on top of each other, and you will get this type of three-dimensional structure. Here are few different representations of such zeolites. Okay, so depending on the amount of aluminum, size of pores varies, and different type of pore size gives different type of products depending on the size of pore. So the reaction taking place in the zeolites depends upon the size and shape of reactant and product molecule, as well as upon the pores and cavities of zeolite. Usually reaction occurs from inside. That means most of the time reactant molecules will be adsorbed from the inside. That means adsorption process will take place here from the inside the pore. Okay, so molecules will be adsorbed at the inner surface of the zeolites, and different type of pore size gives different type of products. As for example, suppose if we assume in this image, suppose molecules are passing through this smaller sized hole, so this will be our product size, and if they are passing through this larger sized hole, then this will be our product. Okay, so these type of reactions or catalytical reactions depends on size. Zeolites can be found in nature as well as they are synthesized for catalytical selectivity. Zeolites are very widely used as a catalyst in petrochemical industries for cracking of hydrocarbon. Cracking, that means when we dug out crude oil from the earth, it contains multiple different type of hydrocarbon chains, and usually the longer ones. So to obtain petrol, diesel, kerosene, etc. from the crude oil, we need to separate out those different lines or different chains of hydrocarbons, and it is called cracking. So for cracking, generally zeolites are being used. As for example, If you want to obtain octane, usually main composition of petrol, then we need 8 carbon chains. So we will select a zeolite which only allows to pass 8 carbon chains at a time, and most of products will be octane. So purity of product will be comparatively high and reaction will be easier. An important zeolite catalyst used in the petroleum industries is ZSM5. So this image shown earlier is a simple representation of ZSM5. In ZSM5, these type of arrangements will be observed. It converts alcohols directly into gasoline or petrol by dehydrating them into the mixture of hydrocarbons. Our next point is enzyme catalysis. That means now we are moving towards the natural catalytical reactions. So enzyme catalysis usually occurs inside the bodies. Enzymes are complex nitrogenous compounds which are produced by living plants and animals. They are actually protein molecules of high molecular mass and forms colloidal solution in water. Now what is colloidal solution? Our next point will be about colloids. Half portion of surface chemistry is about adsorption and catalytical properties, and other half portion is about colloids. So we will discuss about colloids, but simple example of colloid is, suppose in solution we have two different compounds, solute and solvent. If we increase the size of solute, then they are called colloids. Okay, after the certain range it will be called colloids. So how can we identify whether our solution is colloidal solution or pure solution or normal solution? So simple thing is, if solution appears blurry, then it is colloidal solution, and if it is clear, then it is normal pure solution. As for example, if we dissolve NaCl in water, then no change in the transparency of water will be observed. It will appear normal. But example of colloid is milk. Milk is not transparent, or it is semi-transparent. It is not as transparent as water or pure solutions. We will discuss about this topic in detail in upcoming point. So when we dissolve protein molecules, they forms colloidal solution inside the liquids because their sizes are very large. Proteins are very large molecules, so they are very effective catalyst and catalyze numerous reactions, especially those connected with the natural processes. Numerous reactions that occurs in our bodies of animal and plants to maintain the life process are catalyzed by enzyme. The enzymes are thus termed as biochemical catalysts, and phenomenon is known as biochemical catalysis. So our body regularly uses many different type of chemical reactions for survival, and these chemical reactions are normally extremely slow, and we need to increase the rate of reaction, otherwise simple process of digestion of food will take up to a month. That means your food you consume today will be digested after almost 30 days. So these type of processes are extremely slow without the help of catalysts. So many enzymes have been obtained in a pure crystalline state from the living cells. However, first enzyme was synthesized in the laboratory in 1969. The following are some of the examples of enzyme catalyzed reactions. So first example is inversion of cane sugar. So invertase enzyme converts cane sugar into glucose and fructose. So we have cane sugar C12H22O11, that means sucrose molecules with hydrogen. In presence of invertase enzyme, it gives glucose and fructose. Now inversion of cane sugar, what it means? So inversion, that means optical activity of the solution before and after reaction will be flipped. Now what is optical activity? So you may already learn about this in previous units, haloalkanes, haloarenes. So simply when we pass the straight beams of light called plane polarized light from the solution, if solution is optically active, then it will change the direction of light. Suppose before reaction light direction was changing in left hand side, and if after reaction light direction is changes in right hand side direction, then it is called inversion. Before reaction it was changing in left hand side, and now it is turning towards the right hand side, so it is called inversion. So in case of this reaction, that means cane sugar solution, and after reaction glucose plus fructose solution gives opposite optical activity, so that's why it is called inversion. Next, conversion of a glucose into the ethyl alcohol. The Zymes enzyme converts glucose into ethyl alcohol and carbon dioxide. So simple reaction C6H12O6 in presence of Zymes gives ethyl alcohol and carbon dioxide. Conversion of a starch into the maltose. Diastase enzyme converts starch into the maltase, then maltose into glucose. So maltase enzyme converts maltose into glucose, and decomposition of urea into ammonia and carbon dioxide. NH2CONH2 with water in presence of urease enzyme gives ammonia and carbon dioxide. In stomach, pepsin enzyme converts protein into peptides, while in intestine, the pancreatic trypsin converts protein into amino acids by hydrolysis. Now, amino acids contains two different type of reactive groups: COOH, carboxylic group, and NH2. That's why they are called amino acid, because they contains both type of groups. When this amino acid combines with each other, eventually sufficient large chain will be called peptide, and when peptide chains again merges with together and it creates even larger chains, they are called proteins. Okay, so proteins are actually made from, or they are polymers of amino acids. When monomer of amino acids are joined together in a large quantity, you will get proteins. So reverse reaction, that means decomposition of proteins first will give us peptides, and even further decomposition of peptide gives amino acids. Next, conversion of milk into the curd. It is an enzymatic reaction brought about by lactobacilli enzyme present in curd, that means done by lactobacillus bacteria. But this reaction is done because of presence of enzymes in these bacteria. Some important enzymatic reactions are mentioned, listed in this table, so you can study it, and if you find any confusion, then you can ask me.